Let me zoom out then and try and put this in some context. At Stanford, we're posing within the engineering school a question. The question is, how good can we get at engineering living matter? Much like 50 years ago, we posed questions like, how good could we get at programming computers? And we're 10 or 15 years into answering that question, not just at Stanford, but globally. And the early indications are we're going to get really good at engineering living matter. As good as you might imagine as we got with engineering doped silicon semiconductor-based systems. So then the peril, in my mind, is to figure out how we can create and sustain venues where a strategy is realized. A strategy that is needed to determine what it would mean to flourish on top of future biotechnologies, but also a strategy to mitigate some of the negative outcomes. And I'm sure we'll talk about safety, and so I don't want to preempt that, but
Well, yesterday the Recombinant DNA Advisory Committee, the RAC, at the NIH, which was founded back in the 1970s as a result of a prior revolution in biology, the original genetic engineering revolution, approved the first experiment on human beings with CRISPR, which will be conducted shortly. And it is funded by Sean Parker, billionaire of Napster fame. So you see a confluence of different interests here. And the target is cancer cells and modifications that could result in curing people of cancer. So we're on our first big step towards the utilization of this technology in human beings. And certainly we will see some extraordinary breakthroughs that will be very, very exciting in the near future. I would say the thing I'm most enthused about from my own bias as potential applications of synthetic biology, CRISPR, and where it overlaps with basic microbiome research, the other great revolution going on at the same time, is in developing a far broader understanding of pathogens, whether they're viruses, bacteria, parasites, all over the world, understanding the ecological niches they occupy, and having a better appreciation for how we can rapidly diagnose them, how we can assess their likelihood of jumping zoonotically from one animal species to our species or to our livestock, or from one wild plant to an essential food grain of some sort. So I'm very, very excited about where this technology could go in that direction. Of course, like all new biologies, we see that things get dictated heavily by where the stock market chooses to invest. And I don't know if the stock market is interested in preventing pathogens. There may be other priorities that Sean Parker and his pals devote their money to. And that takes to my downside concern. Our understanding of how the microbial world works is so primitive. It's phenomenal how little we know. We have only relatively recently come to appreciate that every human being in this room has far more microbes in you than cells with your own DNA in them. We're only at the cusp of appreciating how those microbes interact with other things around you, around your chair, on that table, in the food you just ate, much less how the same interactions are occurring with animals and plants and so on in the oceans, in the air we breathe, in the soil, etc. I am very concerned that we will have untoward consequences, mistakes made, that result in altering some essential microbial balance that has been in place for millennia and is very vital to some ecological system that we care about. It might be the human body, it might be the body of a dog, it might be soil that we grow wheat in, I don't know. It could be many things. And the reason I'm concerned about this is that, and by the way, this means then that we're deliberately or unintentionally altering evolution, right? I mean, a fundamental change can occur, especially using gene drive, that results in an absolute switch in the evolutionary trajectory. And I worry about this because the technology, especially CRISPR, is so darned easy. You can now buy kits and do it at home. There's a whole DIY movement developing. It's cheap. High school students are learning how to do these things. Drew has played a vital role in enhancing the capacities in high schools. And I think that's fantastic. But I'm worried about what escapes our attention in our hubris, that we think we know the world. And something we do could make a fundamental change.
In a few minutes, we might come.
I'll come back to Drew's approach and really talk about medicine. Because yes, we will be starting the first clinical trials using CRISPR gene editing to modify our body's own immune cells, the T cells, in such a way that they are either more aggressive in attacking cancers or the brakes that have been put on them are lost. And obviously this is of tremendous interest on a global basis and something that could extend what we're currently learning from the new immuno-oncology drugs that have been in the news, the Jimmy Carter drug, for example. But it's just taking that same concept, accelerating it, and allowing us to think about truly novel ways of attacking cancers using our own natural immune system, which is certainly something that should be more preferable than chemotherapy or radiation. So it's very exciting. As Lori said, these technologies are cheap. We'll see an extraordinarily rapid uptake, I'm sure, and a pressure to do more and more. And the peril is obvious. It's safety. It's the specificity of these techniques. Are they only editing the DNA that we want them to edit, or are they actually spilling over and affecting other parts of the body? When you're doing this technique outside of the body, that threat is not so important. But when you're giving this in ways that can be inherited, for example, affecting the germ cells so that the children of treated people are affected, or you can spread ecologically cross-species or into the biome or into the environment in unexpected ways. We have to very much caution our enthusiasm for the benefits with a very humble recognition that there is an awful lot we don't know right now about how these techniques are being spread. And to extend that a bit more globally, right now, for example, in China, the amount of effort that's going into applying this gene editing and gene-derived technology is extraordinary. If you do a Google search for gene editing in China and look at what comes up, you'll see the myriad applications in rice, in silkworms, in human embryos, in various other cell lines and tissues. So on a global basis, we're seeing people run with this technology in environments that don't necessarily have a rack or any of the other mechanisms that we try to use in the United States to balance technology with a safety assessment. And I think that creates a threat, even in well-intentioned people, let alone the people who can adapt this technology for more nefarious purposes. And CRISPR did appear, well, it wasn't named CRISPR, but gene editing did appear on the list of weapons of mass destruction of national interest just very recently. So there is a downside.